Good evening and welcome to TMA's Myositis Research Insights webinar. My name is David Mochel and I'm the immediate past chair of TMA's board of directors. This webinar series is designed to connect patients and their families to specific myositis researchers in our community. Before we get started, please note that this webinar is being recorded and will be available on TMA's YouTube channel on June 1st. Please put any questions you have in the Q&A box at the bottom of your screen. We are pleased to welcome Dr. Kanneboyina Nagaraju, or simply Raju, as I suspect that he will prefer. For those of you who don't know him, he is a very kind, humble, and generous man who has been incredibly supportive of TMA for years. After receiving his veterinary training in India, he attended the Sanjay Gandhi Postgraduate Institute of Medical Sciences, where he received his PhD in immunology and then completed his postdoctoral training at NIH. He is a professor and is the founding chair of the Department of Pharmaceutical Sciences at the State University of New York, Binghamton University, and currently serves as dean. He was awarded the prestigious Distinguished Professorship by the State University of New York Board of Trustees in 2024. In his spare time, he's also co-founded several bioscience companies. Raju is an immunologist specializing in autoimmune myositis. In collaboration with Eric Hoffman and John McCall, Raju is credited with identifying novel glucocorticoid analogs with reduced side effects profiles. Vamorolone is the first drug approved in both the US and Europe for the treatment of Duchenne muscular dystrophy. Gives me great pleasure to welcome Raju, and thank you so much for being with us today.
Thank you, David, and good evening and good afternoon, everybody. And I really want to start by thanking the TMA for giving me the opportunity to talk about myositis research. You know, I have been associated, I have been doing myositis research now almost 30 years. In fact, one of my first myositis research grants were funded by the TMA. And that funding really helped me to not only commit to myositis research, but also continue myositis research. And that is one of the major, wonderful things about what TMA has done over many years, not only bring awareness, but also allow young researchers, I'm no longer young, but at the time, 30 years back, to commit to this particular cause. The more people work on myositis, more newer ideas come. I also stayed on myositis medical advisory board for many years and also chaired the medical advisory board for myositis association some years back. So I really, want to emphasize the role of TMA. I have experienced firsthand myself how much I got benefited. So it is always wonderful, and this is one of the foundations, and I will be forever associated, partly because not only I will explain a little bit about my research in the coming slides, but the support that is afforded to both clinicians and researchers and physician scientists is unbelievably enormous. So with that, as David mentioned, I've been associated with the two companies, and I've been the founder of Rivergen, that is the company that developed Vamorolone, and I also co-founded Agatha Biosciences, working on, again, rare neuromuscular diseases. So I'll explain why I'm interested in those. But I also want to mention that I am not a physician, so it is important that I'm a scientist dedicated to understanding how the disease progresses, how to identify the drug targets, how to identify the drugs, how the immunological interactions occur in this particular rare disease. So I hope you had time to read my disclaimers. So I want to thank my mentor, Dr. Paul Plotz. Dr. Plotz, I don't know, some of you may still know him. He passed away last year. Uh, one of the preeminent rheumatologists at the National Institutes of Arthritis, Musculoskeletal Diseases, worked for almost 50 years solely on autoimmune myositis. And he has trained many rheumatologists, many scientists, many physician scientists. And he was a great mentor, above all, one of the kindest humans who shaped the careers of many, including me. So when I joined 30 years back his lab at NIH, I wanted to work on rheumatoid arthritis. And he told me, rheumatoid arthritis is fine, but myositis is the one where you can actually learn the secrets of nature. So that reverberated and went back to look at this statement by William Harvey in 1657. So the careful investigation of the rarer forms of the human diseases, you learn more than the famous diseases. So that's still today, I continue to work on the rare forms of the diseases so that they reveal its secrets so that we can identify the pathways and interventions and treatments to help patients with the rarer forms of the human disease. So I'm eternally grateful for my mentor for introducing me 30 years back to study myositis, and I continue to study. And I think that's the best advice a mentor has given to a mentee. So why I was interested in myositis? It's very interesting because we have, for those of you who understand, we have lots of proteins in our body. There are, for numbers sake, there are 25,000, not one, two. But for some reason, in myositis, you have autoantibodies to a handful of proteins of the 25,000. That is fascinating. Why on that? These proteins are targets of our own immune system. Because as some of you know well, immune system is designed to fight things that are coming from outside. It is not designed to recognize our own self. So these autoantibodies, they really help us today to associate. If you have anti-MDA5, you probably will have interstitial lung disease. If you have anti-synthetase, you will have arthritis. So on and so forth. But we still don't know whether there is a cause-effect relationship, because many patients actually don't have these autoantibodies. And those who have, they are really prognostic and also very diagnostic, but we still don't know the cause-effect relationship. So, and the other fascinating thing is you all know we have several muscles in our body, more than 600 different types of, but in this particular disease, the proximal muscle weakness. Why this specific unique pattern? So these are the things that fascinated me for many years. So why certain proteins are targets of our immune system? And some of these proteins are also, they're not specific to muscle. They're in every cell in these. There are manifestations pretty much in every other organ, but it starts with the muscle. So I just want you to understand, if you take a tiny bit of a muscle biopsy, how does it look? So this is how it looks. This is what we call hematoxylin eosin staining of a 10 micron section of a muscle. And you can see this mosaic pattern of each individual fiber with own little nucleus on the side. But if you take the same biopsy from patients with myositis, you will see something very different. So just remember this picture, very beautiful picture, no gaps, nothing. And then you will see quite a lot of invading cells. These tiny little cells here, they are our own immune cells. They're not supposed to be there. They show up in the muscle. And here, they are not innocuous. They are talking to each other. They're also talking to the muscle. And if you carefully observe the muscle itself, you can see here is a fiber, very small. In the previous picture you saw, they're relatively uniform. You have huge fiber size variation. Why this fiber is so big? Because it is filling up the empty space by a dead fiber. So what we call this is fiber size variation is common to many muscle diseases, but here what you see, why this fiber is so small. Is it dead or is it dying? And muscle also regenerates. So for the last 30 years, people have specifically focused on how these cells interact with each other, how these cells also interact with the muscle fiber, and how they mediate the muscle death and damage. So that results into what we call so-called muscle weakness. So I wanted to expand on this. You don't have to remember all these names that I will be showing in the next slide, but the concept is as I mentioned, those cells, that little dot things that are invading the muscle, they are depicted here. And as you can see, if you see those arrows, there are almost 24 different arrows. So for the last 30 years, we learned quite a lot about all these arrows, how these cells talk to each other. And we also learned how some of them actually mediate muscle injury, causing smaller fibers, dead fibers. But we learned very little how the fiber itself dies that results into weakness. So my research for the last 30 years has been focused on understanding how the muscle fiber death and injury occurs in autoimmune myositis. So this model that we have been focusing on worked to some extent, partly because majority of the drugs that we develop target those. So let me rephrase this in a little diagram. Here is, you have a nice muscle fiber. So what happens to this muscle fiber in myositis? As many of you know, when we take the blood, you see the so-called serum creatine kinase. It comes up into our blood. That is an enzyme that is supposed to be within the muscle fiber. The reason that is getting out is because this membrane is not stable. So you have the so-called unstable muscle membrane that releases things. In immunology, we call them as danger-associated molecular patterns. That means things that are coming out of the muscle cell, they alert the immune system and say, hey, there is something going on in this muscle. We want you to arrive here, clean up the damage that is taking place. That's what this is, kind of an invitation for the immune cells to show up. So this is what precisely I was showing you in the previous slide. I don't know how many of you see this picture. So these are the immune cells that come in, and they affect the muscle fibers. So during the 30 years of investigations, we identified muscle undergoes quite a lot of things inside. And my lab and others have identified these 3 unique pathways that culminate in damaging our power source. Majority of you have studied in high school, mitochondria produce ATP. Happen 2 things. One is weakness. What is weakness? Weakness is failure of the muscle to develop an expected force. And the other thing that happens is fatigue. What exactly is a fatigue? It is the failure of the muscle to sustain force for a prolonged period of time. So these signals that are taking place from the immune cells, from the muscle cells, they are affecting mitochondria and depriving the ATP, the power that is supposed to be there. And we also know now mitochondria are critical for providing the stability to the membrane. So this process now continues the so-called self-sustaining loop. Now, in this case, you don't know chicken and egg, which one comes first, which one to be targeted. So logically, when you look at this picture that we have, there are so many immune cells that are there that are not supposed to be there. Therefore, get rid of them and somehow we will clean up the disease. That has been our approach. So that is why in the next slide, as you can see this model, there are many drugs developed that specifically target these immune cells because they are predominant cells that are occupying the muscle, and we want to clean up that place. We use prednisone, methotrexate, azathioprine, cyclophosphamide, cyclosporine, IVIG, rituximab. But newer drugs also, if you carefully look at them, they all target these cells. Very recently, some of the wonderful work done by, again, funded by Myositis Association, is now focusing on the muscle fiber itself. So you can prevent the damage, but you also need to grow that immune system. You can eliminate it very effectively. But you need to rebuild the muscle to regain the strength, to reduce the weakness and fatigue. So one of the things that traditionally has been used is prednisone. Prednisone is really a magic drug that really works. How it works, why it is so effective. So prednisone in short term is one of the reduces multiple different pathways. So I'm again showing this complicated picture of leaky muscle activating lot of cells, activating lot of arrows, lot of cells. But prednisone works partly because, see this, it actually affects pretty much every arrow in this interaction. That's the beauty of prednisone. You can get immediate relief, quick relief, but it is a double-edged sword. It can damage other things. So I'll talk a little bit about this part of this. So these three gentlemen won the Nobel Prize. Philip Hench at Mayo Clinic in Minnesota treated a patient with arthritis. At the time, they published a paper in 1948 or so, and within 2 years, they won the Nobel Prize, partly because prednisone is so magical. And the patient who was treated with that drug at the time had energy to go and do shopping in the mall for many hours. So that type of magic that occurred. But the problem is, as I mentioned, the side effects. Side effects are, I don't have to talk about side effects because you are experiencing, many of you who take this drug. But I want to make a point about the side effects. As I can said, I'm not going to read this. They are like one slide full of things, what prednisone can do to your body. And then the other picture gives you little more. So if you look at the side effects of prednisone, it pretty much doesn't spare anything in our body, starting from brain to heart to liver to lung to bone to skin, everything gets affected. So why are these side effects? And this is a beautiful drug that works, but these are all the same problems. So what scientists like me want to do is understand at the molecular level how the signals occur and how good effects are mediated and how the bad effects are mediated. So going back, again, I will show you this little diagram. Again, you don't have to understand the details of it, but understand that, prednisone is nothing but kind of a powerful synthetic stress hormone. That is our cortisol. Except that it is synthesized, it is coming from outside, it is several fold potent. And you all know the effects of stress. It can affect multiple things. So if you go back to your high school, every cell has a cell membrane and it has a nucleus. And here is the little structure of prednisone. These things, they diffuse to the plasma membrane and then bind to these red things, what we call receptors. Now, the red thing that is in the cytoplasm binds to another red thing and enters into the nucleus. So what scientists figured out, if you have two red things, that binds to your DNA, and majority of those side effects which I showed you in the previous picture are mediated by these double red things, what we call glucocorticoid receptors. Now, we also know prednisone works. That is mediated by, see, the single red thing. So the good effects are all mediated by single red thing, and the bad effects are mediated by double red thing. That means, as I was telling you, double-edged sword. That means you have two things on either side. You have a potential to damage anything and everything that come across. But you blunt one side, you have a little bit of a power. It's not a real clear analogy, but I hope you can understand. So this is precisely many people, and also the membranes ligands. So the idea for us is, if it is that simple, why don't we create a structure that only binds to one single thing? That means you can only have good effects if you don't allow this so-called doubling, or what we call dimerization of the receptors. So this is exactly was done now almost 20 years back by medicinal chemist John McCall. He used to be the global head for Pfizer medicinal chemistry. Again, he dedicated his life to volunteering and working with the rare diseases. So what did he do? So this is how prednisone looks. And this is the new compound called, I will refer this with multiple names, but remember, VBP15 is same as Vamorolone, and Agamree is same as VBP15 and Vamorolone. So I apologize for multiple different names. But if you carefully look at these structures, all that he did was he eliminated this tiny little hydroxyl OH group. That resulted into this double bond around between the positions of 9, and that's all he did. And the magic happened. So we do now all these sophisticated assays. So what that does is that creates here, this is deflazacort and this is prednisone. You see these red lines that are 2.8 angstroms that are present in prednisone and deflazacort. They're absent here. That means it can only bind to one of the red things and it cannot bind to the two red things. That is where a tiny small changes can create big, beautiful effects. That's the creation of John McCall. So Vamorolone program, and from the beginning, our aim is primarily, to understand how steroids work, because as you know, it's not only for myositis, arthritis, allergy, asthma, Duchenne's muscular dystrophy. They are used for several conditions. But every condition on prolonged long-term use, you have the same problems. So we spend the 2 decades peeling away these sub-properties and trying to understand to keep and preserve the good effects. And it is like peeling the onion, as you can see, remove the bad ones. So that philosophy, really worked. So I'm summarizing, before we identify these things in the lab, you simply cannot take them to people. You all know that. So we have to test in model systems to see what we are trying to hypothesize is actually true. And one of the very well-known characteristics of prednisone is, stunted growth. If you treat children with prednisone, they are smaller. It's not only children, if you even treat mice with prednisone, and as you can see their body length, this is the prednisone-treated mice, much smaller compared to untreated mice. Here is Vamorolone-treated mice and prednisone-treated mice. So this graph tells you they are smaller, they have stunted growth. Not only they have stunted growth, they also have their bone. You see how small it is? This is how normal bone should look like. And this is much smaller. So, if you treat with Vamorolone at 3 different concentrations, you don't see any changes, but prednisone decreases the bone. It not only decreases the bone length, but also decreases the bone thickness. As you can see, this is how thick normal bone should be, and this is how thin. So that is why fractures, falling are very common if you use prolonged prednisone use. So in these model experiments, we showed that really this particular drug is getting separated in terms of its good effects on the growth, bone length, and bone thickness. But does it still have that beneficial effects? So in mice, we do this experiment called grip strength. As you can see, the grip strength increases with the increasing doses of Vamorolone. So we know we preserved the good effects. We also removed some bad effects or reduced the critical bad effects in animal models. Now, the main thing is it going to work in people? Because that's where we started a company called Rivergen Biopharma in 2008, specifically to focus on developing this drug for Duchenne's muscular dystrophy. The reason is just the prednisone is the standard of care for Duchenne's muscular dystrophy. Many children who are diagnosed at between the ages of 2 and 4, they were put on prednisone. And as you all can see, I know that prednisone causes devastating side effects for these children. Now, over the period of since 2008, it took until 2015 to do all the background work before we moved on to human clinical trial in children with the Duchenne's muscular dystrophy. So there were 6 different trials were done for various phases. First, to look at the safety of the drug at different doses. Next, to look at the safety of the drug in the children, because a lot of drugs are safe in adults, but growing children, it is important because they are not smaller adults. They are unique because everything is growing in them, how the drug affects. So over a period of time, many children were treated, and the trials as of now even continuing in adults with Becker muscular dystrophy. Again, the reason we focused on these at the time was because prednisone is the standard of care. Now, how did it work in these children? So I want to show you the prominent features when the kids get diagnosed with this disease is their ability to get up from the floor. So these children need substantial help. You all can imagine a child between the ages of 2 and 4, they jump like springs everywhere. But the children with DMD, they need to take help. Getting up from the floor, and especially getting up from the floor from a lying down position is particularly harder. Their ability to climb stairs is also a problem. Their ability to walk 10 meters or 6-minute walk test. So these are all the assays that were used to test the drug efficacy. And as you can see here, I'll show you multiple graphs. You don't have to understand. If you don't treat, their ability to get up from lying down position quickly is like this. But if you use Vamorolone, lower dose and higher dose, how fast they can get up. The same thing, their ability to walk, 10-minute walk and run is much better. And their ability to do the 6-minute walk test is also much better. Now, it is all good. They are doing, functionally, they are doing very well compared to untreated children. And also, and as I said, with prednisone, they also do these good things. But the only difference is whether the side effects. As you can see, if you treat these children with prednisone, this is their height. They become very small. And if you are on this drug, this is 2.5 years. As you can see, they grow like normal children. They don't have this stunted growth. But also, another experiment was done. If you look at if you keep these children on prednisone, their height, normally if you are on Vamorolone, the height grows normally. Well, but if you put them on this drug, so this area, as you can see, their height is affected. But if you switch them to Vamorolone from prednisone, they start growing back. So the adverse effects of prednisone, if you they go away if you move them to Vamorolone. Now, do they have mood problems? A lot of children have quite a lot of behavioral problem, irritability to aggression to anxiety, quite a little sleep problems. And again, I also know many of you who are on prednisone also experience. So this drug, at least it appears, again, these are smaller numbers, a sample size of 20 and 30 is relatively small, but they appear to be less behavioral problems with this drug. FDA approved this drug last October 2023 as a treatment for children with Duchenne's muscular dystrophy. There are 2 doses of the drug are used, and 6 milligrams per kg per day, and also 4 milligrams per kg per day. And now some of the children are on almost 3 years on this drug with no stunted growth, no deleterious bone problems, and less mood disturbances. And then last December also, the European Medical Agency approved this drug for same indication, and in UK this January. So all these regulatory agencies approved. But every regulatory agency saw this drug differently. FDA considers this is another steroid, now different from prednisone, whereas European Medical Agency and the UK Medical Agency sees this as a completely new dissociated steroid that doesn't have bone and mood and other side effects. So again, the currently the ongoing research and more data is actually required to go back to FDA to see the differentiation aspect of the drug. Again, this trial was done in multiple different countries. As you all know, Duchenne's muscular dystrophy is a rare disease. And one of the wonderful things is that when we selected the primary outcome measure, we actually asked the parents, what is important for you? And they said, time to stand. Often children fall and, and their ability to get up from the floor as the primary outcome measure. And then we're eternally grateful for a lot of parents. There's not only participation in the trial, this program was, funded quite a lot because we never went to venture capitals, but mostly by several foundations, government, private, nonprofit foundations supported this work. So, as you all know, there are multiple different indications that this drug potentially works. But again, we were not able to focus on other indications, partly because, as David mentioned, Dr. Eric Hoffman, myself, still we do academic research and we still teach students, we still do our own lab research. And, now this drug is licensed to Santhera, and the Santhera in turn licensed to, depending on geographically, Catalyst. So their focus is probably how to expand this. But to help, we have done experiments to show the drug works, at least in the animal models of arthritis, allergy, asthma, and I did some of the experiments in the myositis model. These were not published, but good indications of suppression of inflammation in the muscle. So I will stop here. And again, I want to thank TMA for giving me the opportunity and take questions. And I think, we will take questions next.
Well, thank you very much, Raju. And I think, as you can tell, there's there's a lot that went in. The amount of years, I'm not trying to think how many years was it, Raju, from start? So if you look at it, I think it gives you a good insight into, so from start to, let's say, regulatory approval in the United States and Europe, let's just say, for a certain indication, by the way. I think this doesn't, I think we can talk on that. But I think so, time, how long did that take?
So essentially, from the concept of identifying the pathway, the inflammatory pathway, that was done in 2001. And high throughput drug assays were developed in 2002, and in 2006, then we were able to identify the analogs of prednisone once. And in 2008, we started the company. And then you saw from there, once you identify the lead compound, make sure a lot of compounds, they simply cannot become drugs, partly because they have to be metabolized, they have to be absorbed, they have to be active. So all those things took 20 years.
Yeah. And it doesn't mean that every drug is going to take this long, folks, but I would say it speaks to the process and how challenging it is. So, and then just for clarity's first, so let's say this drug is FDA approved in the United States. It means that it's not approved for everything, correct? Yes. So
it's only approved for Duchenne's muscular dystrophy. It is not approved for other indications.
Okay. And it's an important point. Now, that being said, once you get to this point, it does get easier, to start expanding use and et cetera, et cetera. So the labor is obviously in getting, the novel Well, it's not novel according to the FDA.
Yes. But they still approved. Yeah, exactly.
Sorry, couldn't resist. All
No,
right,
no,
so we
you
have
are
multiple.
absolutely right.
So we have multiple questions which were submitted. Were submitted ahead of time. In addition, we do have some live questions in the chat. And I say, please remember, the focus of this is myositis research. If there's listen, we can answer. We'll try to handle as best we can. Some general questions about medical conditions, et cetera, but very specific conditions would have to probably more appropriate for an Ask the Doc webinar. As an orthopedic surgeon, I would tell you that so part of the long side effects of prednisone over a long period of time is exactly what Raju showed. I mean, there's no question. Now, some of this is dose dependent. Don't kid yourself. How much prednisone? This is where the individuality matters. Okay, if you're on 20 milligrams of prednisone for 20 years, that is different than, let's say, you're on 5 for 10 years, let's say. They are very different, but all of the side effects that he showed there are real. I know there was one of the questions in the emails ahead of time, tapering off prednisone. The short answer is there's not one way to do it, okay, because there's different dosage, there's different lengths, there's different reasons, and that's one of those that's got to be, and if you ask 10 different internists, how to taper off a 20 milligram dose that you've been on for 20 years, you'll get 10 different answers, kind of. But my point in answering that question, which was a very good question, by the way, is it you got to go work with the guy who prescribed it. So,
Exactly.
I would say, so first question, I think let's just stay on the Vamorolone real quick so we could kind of, before we jump off, if that's okay. Obviously, progress with the Vamorolone outside of the Duchenne muscular dystrophy world? What's your viewpoint on where that could go?
Yes. So there are a couple of investigators who are working on juvenile dermatomyositis, primarily at Duke, with support from the CureJM Foundation, started this effort to submit a protocol. And that effort is still continuing, and that is probably one of the first ones if it materializes, because all these, especially academic units doing research, it takes a lot of funding and effort. So, but we do have really committed investigators at Duke who are currently planning a trial for juvenile dermatomyositis.
Okay, thank you. And along these lines, and maybe we can digress a little bit. We get a lot of IBM questions. Is there a chance to research this drug for IBM? I would take a step back and just say, do you consider, as an expert in autoimmune myositis, I guess I think you know where I'm going with this, which is going to be, do you consider, let's say your spontaneous IBM, just an educated guess, an autoimmune disease? Is that is your opinion as best you can tell? Listen, as somebody who's an expert, because there are different opinions whether IBM is actually autoimmune or not. I think clearly it's very different than some of these. So I'd listen. I mean, before we start with that, I would appreciate your input.
No, definitely, Dave. Thank you for that question. Again, it's one of the wonderful things about studying this disease for a long time. We have some history. Yeah. And, when I first, it was in 1995, started reading rheumatology and neurology textbooks, and inclusion body myositis, one of the differentiating factors is not having autoantibodies. Right. So now that is changed. So that means increase the technology, resources, and the availability of samples is making a huge difference. Now, I do think it is an autoimmune disease, and we also have a lot of patients who don't have autoantibodies. But that doesn't mean, because right now, as I mentioned, there are 25,000 proteins. Testing for all of them is not easy. And I also know that in the human disease, antibodies, if they are not there at a certain point in time in the disease, it doesn't mean they're not going to be there in 5 years, 3 years. It is a dynamic process. Therefore, testing continuously. Now, with the available technology, we will be able to identify autoimmune autoantibodies. So I feel inclusion body myositis has clearly an autoimmune component.
Well, thank you. So I think a lot of the questions I'm seeing center into, okay, this is great. When do you think it could be available for when are trials, going to be available for, DM or PM or antisynthetase, et cetera, obviously, in this country, in Australia, et cetera. So put it like this. It's a challenge to try to predict, but your best guess as far as, okay, so at this point, the world is focused on children, right? And Duchenne muscular dystrophy, right? So what's the process, if you could explain to the audience for, it's not just a simple matter of, okay, we're going to try this now, we're doing a DM trial, on that. So what's your best guess as far as the path forward for looking at these other diseases?
Yes, I think it is in the best interest of these companies to work as many diseases as possible to be on their label.
Yeah.
Partly because, theoretically, this drug should work wherever prednisone works. So that process, how long it takes, it all depends on their ability to raise resources to do these trials. And it is a complex process. How they prioritize depends on, there is some business decisions. And also, I think what worked best in my mind is the push from the foundations, the need, because FDA really listens to the foundations and organizations and the need, especially myositis is a rare disease. So by doing research on rare disease, FDA handholds these companies. So using those benefits. So in terms of the real, how many years, I really don't know, but I know this much, having a lot of friends within the myositis research community who are doing clinical trials are actively trying to initiate protocols to test this drug in myositis. But how long it takes, it again, individual investigator initiated trials, they're already overrun with other trials. Now you have to add these, how long that takes and how much resources that they can secure.
No, appreciate it. So I think to all the questions about when are trials coming, when can I sign up? When can I do this? Where can I get this in the United States? The short answer is you can't get this right now in the United States for myositis. Is that correct? Is that a fair statement?
So I think it is, the company cannot sell this
Right.
drug for myositis. But I don't know, you are a physician, much better about this, whether physicians can prescribe. So, but the company cannot sell this drug. No company, because it's not on the label, it is only for Duchenne's muscular dystrophy.
Yeah. So it doesn't mean that it can't happen. And this is where actually the fact that the FDA doesn't considers it another type of prednisone may actually be helpful. So, so we shall see. No, thank you. Appreciate it. So a couple of some good, so I think good thoughtful question. All right. So have you examined differences? Okay, here you go. Examine differences in gene expression in any model, Duchenne's myositis, between prednisone and Vamorolone. Have you examined differences in gene expression in any model between prednisone and Vamorolone?
Yes, we did it. So as I mentioned, so it induces significantly less number of genes. I should say not only induce, so this goes upregulation and downregulation. So it affects significantly less genes compared to prednisone or deflazacort.
Thank you. For those on the call, just as more of a clinic, dermatomyositis and let's say now muscular dystrophy is a broad category. Is dermatomyositis and muscular dystrophy related, would you say, from your standpoint, what similarities do you see in those two? They're both muscle diseases, right? But
Right. No, exactly. So a very good question. I think the Duchenne's muscular dystrophy is caused by the absence of one single gene, that is dystrophin. Whereas dermatomyositis, we do not know. These are often autoimmune rheumatic diseases are often called as multigenic diseases. There are no real mutations that cause the disease, but there may be single nucleotide polymorphisms in multiple different genes. That's why they are called polygenic diseases. It's not more than one gene. Whereas Duchenne's muscular dystrophy is caused by one single gene. So it affects male children partly because the gene is on X chromosome, whereas dermatomyositis, we do not know what causes. That is part of some of the work that I did for many years, identifying the susceptible factors that cause dermatomyositis. And the other component is the muscle inflammation is common between these two diseases. That is why Vamorolone is likely to work in dermatomyositis, but it is not same as Duchenne's muscular dystrophy, partly because the single gene effect.
Got it. All right. So your best guess as far as chicken or the egg, looking at the cellular level, where do you think? Yeah. Do you think it's an intrinsic? Where do you think what's your best guess as far as what starts the process of the muscle damage?
Right. I think in my own guess, it's a guess. Again, please don't quote me.
Disclaimer here to the crowd, but if there's anybody who could provide a good guess, I would be interested to see what you say.
So I think we all have susceptible genes, and we are not, especially with myositis, you're not born with this disease, unlike Duchenne's muscular dystrophy, because that's a genetic defect. So when you have susceptible genes, all that you need is a trigger. That trigger can be anything. It doesn't have to be. So people try to identify a virus that causes myositis. You never found a unique one. What you found is like any other virus that is present in general population is also present. That means how this virus unmasks the genotype of a susceptible background versus a non-susceptible background. For example, these rare diseases, they affect less than 0.1% of the population. So if the virus is not going to be unique, it is the same virus that affects everybody else, affects people who are susceptible to myositis. Only thing is, in everybody else, virus comes and immune system eliminates, virus goes away, and then that fatigue and other things, we are back to normal. But if you have a susceptible background, that trigger is what initiates and continues. So inability to go back to normal is lost in most of the autoimmune diseases.
Appreciate it. Last couple quick questions. Specifically speaking to impact of liver and weight gain with Vamorolone versus, meaning side effect pattern similar, or let's specifically focus on liver function and weight gain with Vamorolone versus prednisone.
So some of the weight gain, a subpopulation of children did gain at high doses with Vamorolone, but it is not exactly the same level as prednisone. It's only a subgroup of kids. Again, in them, how the drug is being metabolized.
All right. I think, look, the last questions. I would say, and I appreciate you mentioning the FDA listening to patients, probably as a patient advocacy organization. Not going to lie to you, it's nice to hear that as somewhat of a skeptic when it comes to government agencies and these things. Raju, I would say that's very helpful, and it's important that came up. So I think this is more of a question for Paula. It was a great question submitted by one of the attendees, and that's it's more focused for TMA and us. And so we are in the process of multiple efforts along these lines, but I think this will just has us double down, move forward. So I know I didn't get to all the questions, but I would say last couple good questions. It's anti-inflammatory diets, that sort of stuff. I think we can maybe focus on put it like this: if there was one food we could eat that would cure these things, trust me, we would eat it. But I think it's the hard part is it's listen, as a physician with the disease and just watching myself over 10 12 years, seeing how everybody is so different, I mean, it is stunning. And there's just not one. Put it like this: it's a rare disease and it's not understood for a reason. Okay. I mean, that's the bottom line. These things are just, hopefully at some point, St. Peter, when I'm in purgatory for will tell me, okay, it was when I sprayed mosquitoes and was, for that one summer and was, exposed to all those chemicals. That's what it was. It probably won't. It'll be something else, something in the cushions, that a virus I had when I was 6 that probably was the cause. But hopefully we'll find out someday. So I apologize for those who didn't get to the questions. Go ahead, Raju.
Thing, my general advice is, so physiologically muscle, we should not keep our muscles to be rested. I think it is important to move, to do what you can, not to exert, but activity is critical. And exercise, and again, I know TMA has quite a lot of things on their website in the resource. Physical exercise and non-pharmacological intervention is one of the best for the muscle, partly because, as I mentioned, exercise helps mitochondria. Mitochondria helps everything else. So my own suggestion is, please try and be active to the best of your ability. There are experts on the Myositis Medical Advisory Board. There are quite a lot of world-renowned physiotherapists who can help you what is the best exercise. And keep moving, keep being active I strongly believe, is the best effective intervention for this disease.
Could not agree more. And on that note, Raju, once again, thank you for your expertise, generous time, and we're very fortunate to have you as part of TMA. Thank you so much. Really appreciate it.
David.
And thank everybody. We had a fantastic turnout. I apologize we didn't get to all the questions, but recordings will be posted in the YouTube channel June 1st, I believe. So to everyone who tuned in, thank you so much, and have a great day.
Thanks. Bye.
Bye-bye.